this animation is showing or illustrating how we're going to modify C3. And the first thing that happens is we're going to generate from C3, in the presence of water, a complement component called IC3. The complement pathway components include C3, factor B, factor D, and piperidin. C3 cleaves into C3b and C3a. We also generate some IC3b, and we're going to find that just simply having these fragments of C3 floating around in very small concentrations in plasma is enough to get the alternative pathway started. This is a magnesium-dependent process, and as C3 undergoes spontaneous hydrolysis in the presence of water, it's going to interact or bind to factor B. Factor B is then going to be cleaved by factor D into, again, a large and a small fragment, BA and BB. BA is the small fragment, floats away, and does have a particular immune function. The large fragment of factor B attaches to the hydrolyzed C3 and attaches to, essentially, the cell surface. That C3 with water and BB complex is a C3 convertase. It fairly quickly starts to cleave more C3 into C3a and C3b. C3b is going to be inactivated into IC3b if it's simply floating around in the presence of water. But if it's close enough to the cell surface of the bacteria, the C3b all by itself will attach to the bacterial cell surface, as well as eventually the whole C3BB complex. So here again we have the illustration of C3, and now with essentially an interaction with factor B, we can start to generate the C3 convertase of the alternative pathway. Factor D is going to function like a pair of molecular scissors. So the function of factor D is to, again, cut B, factor B, into a large and a small fragment. And that's the C3 convertase for the alternative pathway. The C3b-BB deposited on the bacterial cell surface. On the bottom left, we have an illustration of some of the regulatory proteins that can control the activation of the alternative pathway. You don't want random activation of complement or the complement components for any of the pathways. In fact, all three of the complement activation pathways are highly regulated. Multiple steps in the process. Whenever you see any biological process that's performed in a variety of tiny little steps, always think in the back of your mind, oh, this is a highly regulated or controlled process. We want to very carefully, the process has to be naturally very carefully occurring, so you don't have random or erroneous activation. You want to have this process occurring when you really need it. So we see on the right-hand lower image, or cartoon drawing, where we have the appropriate proper activation of C3b-BB, the C3 convertase for the alternate pathway. Once we have that C3 convertase formed, an additional protein called preparedin, or factor P, comes in and stabilizes that C3 convertase on the bacterial cell surface. So preparedin, or factor P, is going to, again, make sure that enzyme remains fully functional and intact where it needs to be. If we find that C3b binds to some of the inactivation or the regulatory proteins, then we won't get further activation of complement, which is illustrated on the left. What we're focusing on is the actual appropriate proper activation for the alternative pathway and the formation of the C3 convertase, which is C3b-BB, on the cell surface. We'll also have further cleavage of more C3 into large and small subunits, with more C3b deposited on the bacterial cell surface, which can serve as an opsonate. In addition, some of the C3b will attach to the C3b-BB, C3 convertase, and change that enzyme function to a C5 convertase. So here we have a chart that's comparing the various different types of proteins and their functions in the alternative, the MBL, and the classical complement pathways. For initiating serine proteases, factor D is going to function that way. For MBL, it's going to be MASP, and for the classical pathway, it's C1s. For covalent binding to the cell surface, for the alternative pathway, we're using C3b, but for all other pathways, we're going to use C4b. To create the C5 and the C3 convertases, for the alternative pathway, we're going to use factor BB, in other words, this large fragment. But for all other pathways, we're going to use C4b and 2b. For opsonization, C3b serves as an opson for any possible pathway. And once we've generated the C5 convertase for either the alternative or all other complement pathways, then we're going to cleave C5 into a large and small fragment, with C5b serving as the initiator of the membrane attack complex. C3a, C5a are the primary inflammatory fragments that are produced when complement components are activated. C4a has weaker inflammatory action. C2a is going to go off and actually interact with another component called bradykinin. And so, again, nature doesn't waste any of these parts. In fact, factor BA serves in other pathways as well. So here we have an illustration or comparison of the C5 convertases of the MBL and classical pathway on the left, including CRP activation. And then we also have on the right the alternative pathway, C5 convertase. Each C5 convertase is going to cleave C5 into a large and a small fragment. It still has the same enzyme function, it's just structurally these are different in terms of structure, but the function is the same.